can do, a million, different, analysis at a time, which then, you kind of, put together into a genome, right? And then this notion that you could do the same thing with cells, right?
Yeah.
This is, novel and imaginative, right?
Yes, absolutely, and I couldn't phrase that trajectory better. I think the perspective I would share here is that there are probably like three aspects to it that resulted in this massive scale up, right? One is that you already mentioned the fact that you could just take pictures of cells and they have rich information in them, and you can read out not one thing but hundreds of things, and we'll come back to that. But then there's also this other thing of robotics and of high throughput imagers that are just able to record things at scale very, very fast. And the field of high throughput imaging, where you can basically take these, for the listeners who don't know what multi-well plates are, these are essentially, you can just think of them as probably the size of a large iPhone or something like that, a small tray with really small test tubes. And in each test tube, you can do a separate experiment. Test tubes are called wells. And so you can do all these experiments, and these are typically the workhorse of high throughput anything, high throughput imaging included. And that technology of just being able to do experiments in high throughput, that sort of had started picking up pace. And then separately, from the point of view of perturbations, we've always had large chemical collections, I guess. But you know this better than I do, Javier. Now it's possible to do that first in silico and then actually go and produce the chemicals that you care about. I don't know too much about that field as much as you do, but there's also this genetic perturbation aspect, right? It
Mhm.
became possible with CRISPR, but also preceding CRISPR ways, high throughput ways of really scaling up that.